Thank you, Chairman, for kind introduction, and also thank you for inviting me to share with you our experience in fish in Hong Kong in last 20 years. As you know, the leukemia diagnosis, we need a very good hematopathologist, have a good morphology for have a bone marrow smear, and also we need to have immunophenotyping and also the cytogenetics. And also we need also with a molecular cytogenetic fish and also molecular tests to have a very good accurate diagnosis for the leukemia case. And this shows you the genetic analysis is very important. As you know, the WHO is also classification by genetic aberrations, such as they have a very good correlation of the survival of the genetic aberrations. However, very important of the cytogenetic karyotyping for the leukemia case, there are still a lot of limitations of the karyotyping, such as we need to culturing the cell is require the living cells, and also the metaphase size is very important because we need to have a very long time to analyze each chromosome one by one. And also because of the cancer cell, the morphology of the chromosome are very not so good, and therefore the resolution of chromosome band are not very good in the cancer cells. And it requires the considerable expertise and very labor-intensive expressed only for the AML-M6. I have an experience for we need to sign a case for a week. And very expensive for the test because the salary in Hong Kong is so high, in Hong Kong. And therefore, the scientists will develop molecular cytogenetic methods. We call it FISH, for instance, in situ hybridization. Simply, it is just the use of DNA probes to detect the changes of human chromosomes, just like molecular biology techniques. And the basement DNA with a complementary DNA probe with a conjugate with a follicle, and with covalent bonds. And therefore, if they have a complementary to the basement DNA, they will have a fluorescent signal. It's very simple. The advantage of FISH analysis, which can be detect the genetic aberrations measurable in the dividing and also non-dividing cells. That means we can detect the interface and also applicable to many specimen type, and no need to culturing the cell at this time. And it is very good correlation with the cytology and also immunophenotyping. I'll show you later. And the test basically is very rapid, maybe 1 or 2 days you can sign the report. And very good sensitivity and also very specific to the gene rearrangement. And for the FISH in hematology cancers, it can be used for detection of the numerical and also structural aberrations in interface and also metaphase cells. And it can be characterization for the marker chromosome. What is marker chromosome? Marker chromosome is an unidentified chromosome by a cytogeneticist. And also it can be detection for the cryptic rearrangement or novel rearrangement of the genetic 2 chromosomes. And also it can be used for lineage involvement by the cancer cells, the cancer clones, and also can be used for detection of the gene amplification, the oncogene amplification, and very good for use for disease monitoring after treatment. And also very good for the use for the chimeric study after post-sex mismatched the bone marrow transplant. This shows you the figures which we are always used for detection of the chimerism of the post-BMT after mismatched BMT, and we are using the centromic XY FISH probe for on the peripheral blood. It's more non-invasive compared with taking the marrow. And here show you the XX is a male, it's a female. It's a XX with 2 dots of the red in color. And here show you the XY FISH. And here this the red dot is a X and the Y is a green in color. And this is a male, and the sister donate the bone marrow to the brother, okay? And here, when we are using the peripheral blood, we can counting the cell is around 10% of the receiving cell, 20% of the receiving cell, and then we keep closely monitoring the patient by using the peripheral blood, using the FISH method. And here show you there is a correlation of the cell lineage involvement using the FISH and also cell morphology. We can correlation the FISH, the signal pattern, work together with the right skin cell, okay? And then we can relocate the area that the cell we are involved. For example, there's a blood cell with a trisomy 8, okay? It's a Down syndrome patient with an additional chromosomal 21. And here show you the neutrophil are not involved, okay? The blood cell are involved, and also the lymphocytes are not involved, okay? Correlate with the morphology. And therefore, FISH are very good for correlate the morphology together with FISH method. And as you know, the multiple myeloma, there is sometimes the plasma cell is not so high in the male blood, and therefore we can using the cytoplasmic immunostaining together with FISH. For example, we are always using the 13q14.3, there's the deletion, and there's a plasma cell, there's I think it's a lymphocyte, and here you can see there is a loss of one signal on this plasma cell, and therefore we are together with the FISH and also cytoplasmic immunostaining, we can correctly identify the plasma cell, okay, the CD1, the multiple myeloma cells, and also we are also doing the FGFR3, okay, there's a two rearrangement here, okay. And also the P53 deletion and the centromere probe in green color, and the loss of one of the red color in these figures. And then how to choose the FISH probe for the detection of gene rearrangement. Normally in hematology, we have two set of probe set. The first one is the dual color translocation probe we used for the lung partners. And also the other one is the bigger parts, which are for the unknown partners, such as the ML rearrangement. And there is a development is very good of this kind of FISH probe. For the development of the BCR label, we can have a fully development of the fourth generation of the FISH probe. The first one is the single fusion. The second generation is extra signal, and the third one is dual color dual fusion. And the fourth color is the tricolor. That means there are three kinds of color, including the ASS gene on the chromosome 9q34. And for the single fusion probe, we have for the negative pattern is 2 green, 2 red, okay, or orange, okay. We have ABO gene on this, okay, in this color, and the BCR gene in green color. If there is a positive signal, we have here the positive signal here, okay, the fusion signal, and we have one fusion. However, the false positive rate is very high. As you know, the cells are three-dimensional, and therefore the cutoff may be 10% to 12%. And therefore, if you have monitoring the patient using the FISH, there's a problem, okay? If you are doing the diagnostic, there's no problem, because for the CML, I think over 90% is a positive cell. But for the monitoring, there is a problem. Is the patient just with a 9% or 8% cell of in the blood, and therefore the scientists will have the other strategies to extend the signal on the Q arm, or on the 9Q arm, or with an extra signal. That means if we have the extra signal, and therefore this is truly positive. If we without the extra signal, that means this cell is a false positive cells, okay, and therefore we count as negative. And therefore the false positive rate will have less than 2.5%, okay? And then we still have a pitfall for the less than 2.5%. That means if the patient with a 2% positive cell, and therefore, how come? Maybe there is cutoff is we just report the patient is negative. And therefore, the scientists will extend at the end, okay? with an extra fusion. That means you have got two fusion signals. That means there is a positive cell. And at that time, the false positive rate is very low. It's less than 1%. Normally in my lab, we count 300 cells for the after treatment, the patient. We count 300 cells if you pick up one cells, and that is also positive, okay? However, the FISH probe development are very good, but for the dual color dual fusion translocation probe, but there's still some problem, okay? For example, in CML case, we still have around 10% of the patient with able BCR deletion, okay, on the derivative chromosome 9. And there is, of course, we can using the single fusion to rule out this deletion at the diagnostic marrow, and we find that maybe the deletion of the BCR able on the or able BCR on the derivative 9 is occurred at the time of Philadelphia chromosome translocation at the same time, and normally the deletion of the 9q deletion is predicted for a poor prognosis in CML. And here show you the case. We still have a 922 translocation on this, but using the dual fusion, we have only one fusion signal, and therefore this is not as a criteria as recommended by the FISH probe company, and therefore we cannot say that this is a positive, okay? However, during the diagnostic marrow, there are over 90%, okay, of the cell with this kind of signal. And we can also sign the report using the single fusion to document that the Philadelphia is still present on the patient, okay? And when we are using the metaphase FISH, we can document the chromosome 9, the normal line with this signal here, okay? And here the normal chromosome 22 are in green, and there is a Philadelphia chromosome, the fusion signal in derivative chromosome 22. However, the derivative chromosome 9, there is a loss of signal on this area, okay? And therefore, the scientists developed the other kind of probe. We call it 9q tricolor, okay? They also with a FISH probe in aqua, in blue color, okay, on the SS gene, on the chromosome 9q. At this time, if we have a phi filter, okay, the one is a B-able in red, the BCR in green, SS in blue, okay. If there is a negative, the blue will fuse with the red color, okay, and this is a normal, okay. For the BCR fusion, we have the two blue dots also on the SS gene, okay. But however, on the BCR-able fusion and the able BCR deletion, one of the blue dots are lost, okay? And therefore, the Philadelphia here, and there is a loss of the blue dot. And therefore, this is a true 9q deletion case. And if we are just using two color without the blue color, and therefore, how come about this cell? If we have a color, to add one more probe on the 9q, okay, at the SS gene, and we have a two dot here, and that means there is a false positive. It's not the true 9q deletion case. For the I also show you the very interesting case is an encrypted insertion of the BCR-able at 9q34. For example, for this CML case, we still have a normal karyotype. The 29 and 222 are normal. However, doing the interface FISH, we have a very fine band, okay, the green signal on this red signal. And doing the FISH, the metaphase FISH, we can find that there is a normal 22 are there, and the 22 are here, and the normal 9 here. However, on the derivative chromosome 9, okay, one of the normal line will have a fusion signal, and this is a insertion, okay, it's just like a sandwich, and BCR are take out, okay, and then insert to the able and become a sandwich to become an able BCR able, okay, and then the BCR able are still expressed. And here I show you we are also using the telomere 1 gene fusion, okay, for doing the ALL case of the childhood. We find that there is around 20% of the childhood ALL harbor the TLML1 fusion transcript, okay, in our institute. And we find that there is some of them show the extra copy, and some of them with amplification of AML1 gene, and some of them show the deletion of tau, normal tau. And also some cases show the duplication of TLML1 fusion, and also some of the case with the loss of normal chromosome tau together with the duplication of the derivative chromosome tau, translocation tau 21. And therefore, maybe additional or secondary genetic changes, including the AML1 amplification, are commonly encountered in the childhood ALL with TLML1 fusion, and this is these genetic changes are expected to play very important roles in the disease progression. And here show you we are using the FISH probe. It's an extra signal. If the positive cell for the TLML1 rearrangement, there is one more AML residue. Okay, there is an extra signal probe. And for the case, we have the case one, we have a AML1 fusion together with the extra AML1 signal. There's a fusion here. However, in this case, we have also found the cell without the fusion, but with AML1 triple point, or the AML1. That means 4 copies of the AML1. And this is maybe the tetraploid or 21 of this AML case. And case 2 with the TLML1 fusion, okay, and also with the cell without AML1 fusion. And we are using the centromere probe 12 in green, and also the orange in the AML1. We also find that there is extra copy of the 12, okay, the normal 12. And the case 4, there is an AML1 amplification. You can see here there is a bunch of the red dot. That means there is AML amplification. When we are using the CGRH method to confirm this kind of case, we find that there is an amplifier on the 21q. And here is also using the AML1 FISH. We can confirm on this chromosome there is an amplification of the AML1 gene. And the K5 we have with TLML1 fusion, and also with AML1 residue. And however, there is interestingly is a loss of normal tau, and some of the case with an extra signal of the AML1 residue. That means there is a 2 copy of the residues. So when we are using the whole chromosome planing probe, we confirm that there is a loss of the chromosome, loss of the normal 12, but duplication of the derivative chromosome 12. And here show you when we are using the CGH, we find that there is a loss of the telomere end of the chromosome 12, the tau gene. And for the 6th case, there is a TLML1 fusion. There is 2 copies of the fusion, okay? And chromosome 4, there is a TLML1 fusion and with a normal tau deletion. And when we are confirmed with a centromere probe 12, they still have 2 copies of the chromosome 12. That means there are 2 copies of chromosome 12, but loss of the tau gene, okay, on one of the chromosome 12. And why I show you this case? Because I want to show you, using the FISH, we can also detecting the genetic clonal evolution in childhood ALL with TLML1 fusion. For example, normally they have a normal clone, and therefore they have evolved to the chromosome trisomy 12, and then some of the clone with trisomy 12, and some of them with trisomy 21, some of them with trisomy 12, and some are clonal evolved on chromosome TLML1 fusion. And some of the case they have the first event is AML1 amplification, and then with TLML1 fusion, and TLML1 fusion, and then they further step to the duplication of TLML1 fusion, and some of the loss of one of the normal 12, and the duplication of the derivative chromosome 12, okay, and some of the case with a TLML1, and then also with clonal evolved to the trisomy 21. And is that the FISH method is good or the conventional cell genetic is good? We have also reported some case with 15, 17 translocation, but not associated with APL and negative for PML RARA. You can see here the PML RARA. And therefore, if you find that this is an M2 case, okay, AML M2, and therefore, if you find that there is not correlate with the morphology, it's better to go to do the molecular test, such as the FISH. And also, is this a FISH is good or molecular technique is good? The PML, for example, for this case is the Philadelphia positive CML with the BCR-ABL variant transcript. There is the 922 translocation is the CML morphology, and also there is a FISH positive for the BCR-ABL. However, we find that the case is not the P190 and also not the P210, but it's a variant span here. When we cut out the band and sequences, we find that there is a E19 and A2 BCR-ABL fusion transcript. That means there's not the P190 and also not P210, the CML case. And the FISH is no global view of the chromosomal compartment and also requires the pathological information and also cytogenetic information before you request this test. However, FISH is manageable by the routine diagnostic lab and also can be answered the specific question for the clinical question and supported by the practice or the personalized medicine. I think is very important nowadays. And also its practical advantage such as numerical aberrations. The multiple fusion partner can be documented using by FISH, and also the breakpoint heterogeneity are also confirmed by the FISH. And it can be applicable to many specimen types. And FISH is a symbol of the health and wealth, okay? And therefore, in Chinese, 鱼象征健康与财富, okay? And thank you very much for your kind attention.